So let's read the question and see if it makes sense. So the first one is the position of a gene on a chromosome. So basically, if I'm just making it as a block. If we want to change, so this little piece there, so that little segment would be called a locus. Sort of horrible drawing, but as long as you understand what we're trying to get to. A single unit, so again a single unit, so one piece of a nucleic acid. Also in understanding that we have two nucleic acid, DNA and RNA, but basically what we want to ask is what is a small nucleic acid, right? So that would be a I forgot to write alphabet. So the first one was E. The single units of a normal nucleic acid, nucleotide, that's a G. A random change, so that's kind of nice. A random change, a random mutation that would happen in the DNA sequence. So we need a word. Oh, there, mutation, a mutation. Yeah, so basically a change is a mutation. So if you take a piece of what would be reading something and you kind of change it. A molecular produced from photosynthesis. Obviously, we understand that photosynthesis makes protein, right? The protein synthesis making proteins. So we would want to amino acid. Produced from photosynthesis. No, not amino acid. Protein synthesis. So amino acid is one building block. It's not the molecule formed. So maybe A would be closer, but we'll see if amino acid is one. It wouldn't be okay. Yeah, so I'll say polypeptide because that's a chain. Okay. A biologist who conducted experiments to investigate the inheritance of traits. This one's kind of nice. So where we kind of get our traits from, or what we believe certain things to be, would obviously be George. Gregor. A scientist who discovered the structure of DNA. That would be James Watson, definitely. See? The nitrogenous base found in RNA. So the nitrogenous base. So nitrogenous base is the things that would change. So obviously we would have like thymine, cytosine, guanine. So which one would be found only in RNA? And that would be uracil. So that would be J. A single unit of a protein. Yeah, that's better for H, for an amino acid. An enzyme that joins two DNA molecules. An enzyme. So it's like a bond. Like what would bond these two molecular DNA? What would bond two DNA molecules? So we know we have hydrogen bonds, but those are bonds and not enzymes. So an enzyme actually ends with an A's. So DNA ligase. A technique that increases the amount of DNA in a sample. So we just want to increase DNA size. So it's actually PCR because that's the last one that's left. So someone let me know what that means. So am I just